Hello everyone, Dr. West here. In this lecture we are going to begin to talk about your sensory systems, and we are going to be walking through sort of each of them individually. At least at first we're going to talk through vision. You can have a couple lectures on vision and a couple lectures on the auditory system, and then you'll see that we're going to start to kind of squish them together a little bit. We'll do some of them sort of together. The reason for that is twofold. First of all, that's sort of how your book has them lumped together. The second reason for that is because that is sort of what we know about your sensory systems. We know a whole lot more about your visual system and your auditory system than we do about some of your other sensory systems. We just haven't studied some of the others quite as extensively, and we just don't know as much. We haven't dedicated as much research time as we probably should have to the other sensory systems. And there's a lot of reasons for that. A simple one is just because we don't use a lot of those other sensory systems, or at least we don't think we use a lot of those other sensory systems as much as we do. But nonetheless, anyway, you're going to hear in the next couple lectures a little bit about some of the sensory systems. But before we really jump into that, we need to talk about something we've already touched at this semester, which is sort of the job of your nervous system, particularly the sensory neurons, and why you even have them and what they're doing. And then we're going to talk about sort of an overall pattern of what's going on with sensory information as it's being brought into the nervous system. So let's talk about this idea here for just a minute of sensation versus perception, because these are two different words here. And what I want you to understand is it almost sounds like a philosophical thing rather than a psychology thing, but it really is a thing, right? Our brains only receive a small fraction of what's actually happening out in the world, right? We're only getting a little tiny piece or tiny snippets of information of what's really going on out into the world. And the way that we actually get that information is because we have various kinds of sensory neurons that have somehow detected energy out in the world. And again, that can be in our external world or it can be in our internal environment. For again, something like, your stomach hurts, something like that, right? You've gotten some kind of sensory neuron that has been activated. It has converted that energy into an action potential. We talked a whole lot in Unit 1 about what action potentials were, how action potentials were sent, how they couldn't jump from neuron to neuron because that would be kind of like lightning. So to get from neuron to neuron, they had to send a neurotransmitter. Hopefully that neurotransmitter was able to then activate, right, the next neuron so that it was stimulated enough to reach threshold. And if it reached threshold, it would also produce an action potential and keep passing that message on so that it would reach the nervous system. Right? And that's eventually how all that information would get to our nervous system. So what we're going to talk about in this particular series of lectures, and not just this one, but this whole sort of unit, we're going to be talking about the sensory systems, is how exactly does that happen? How do we have these specialized neurons that we call sensory neurons that can take that energy that's out in the world, such as light in the case of our eyeballs, and turn it into neurons? Right? How is it that we do that? How do they have that special ability? And then, right, what are the pathways that then go from, for example, our eyeballs into our brain, or from our ears into our brain? Right? And then once it gets there, how does our brain then interpret basically what's just incoming action potentials and incoming transmission of energy from action potential and neurotransmitters, basically from the signaling of neurons, into what ultimately you're going to experience as, oh, I heard a noise, or even that, I heard a specific noise of a dog barking, or I smell a specific smell of cookies baking in the oven. Right? So that's where this idea of sensation and perception comes in. So here's kind of the formal definitions. Sensation is really kind of the boring part, although it's really super important. Sensation is just the work that is done by the sensory receptors, the sensory neurons of sensory organs, to convert the external energy into an action potential. So when we talk vision, you're going to hear about you have these specialized sensory neurons in your eyeballs that literally convert incoming light, and when that light hits it, it produces an action potential. Right? And ultimately that's sent on to the brain. You're going to hear later in a couple lectures from now how incoming moving air will go and stimulate your ear. Right? It will actually cause a membrane to move called your eardrum or tympanic membrane. There's a couple little bones in the middle that move. It's going to tap on an inner part of your ear and make some fluid move. And then as that fluid moves, it's going to move these little hair cells around. And as those hair cells bend over, that's going to cause an action potential. Right? That part is the sensation of literally light, moving air being translated into moving water, causing an action potential. Right? As a chemical lands on your tongue, that chemical will stimulate a receptor on your tongue that sends an action potential. Right? That is the sensation part. Sensation doesn't mean anything unless that action potential that it produced gets to the brain, and the brain is then able to interpret it. So perception is what happens in the brain, where the brain interprets it. And there's a lot of layers to the interpretation. So the first thing that the brain interprets it is what kind of sensory information is it? Is it a sound? Is it a smell? Is it a taste? And then it probably adds even extra layers, right? What specific sound is it? Is this a dog versus a cat? Is this cookies versus cake? Or is this cookies versus spaghetti? Okay? So all of that stuff is perception. So really the only part that you consciously experience is actually the perception part. But you wouldn't be able to have the perception part if the sensation part didn't happen. So you need the sensation part. You need the special receptors that are able to convert the energy, the sound waves that are coming into your ear, the light energy that are coming into your eyeballs, the chemicals that come onto your tongue, or the chemicals that get sniffed up into your nose, stuff that touches your skin, right? And convert it into an action potential. Because if it doesn't get converted into an action potential, it's not going to get sent to the brain. And if it doesn't get sent to the brain, you're not going to be able to interpret it as anything. And if you don't interpret it as anything, you're not going to experience it. Right? So really, our visual experience, which is the one that's written here, but really any experience, our auditory experience, our taste experience, is really not exactly what's out there in the world. It's actually a reconstruction of the information that we got about what's out there in the world. So it's very limited to only the pieces that we actually sent information about. In other words, we experience the world as we need to experience it. At least hopefully we do. Right? Because right now, there are other sounds that are going around in my house, right? And outside of my house. Like I think a neighbor's mowing the grass. If I listen hard enough, I think I can hear a lawnmower going on. But I don't really need to experience that right now. So for the most part, that's not going in. Right? But my daughter is home today, and she has a friend over. And I can tell you that if I heard either one of them screaming or making some sort of noise happen right now, hopefully that information would go immediately in, and I would be able to perceive it very quickly because I would need to experience that. So basically, there is some stuff that's not going in right now. And there are probably lots of other sounds and things that are happening that I'm just not having going in because I am not sensing them. Right? There are literally action potentials that are not being sent because that information is not going into my ear enough to actually make an action potential happen. So therefore, I am not perceiving them. So I don't know what's happening out there in the world. Right? But again, hopefully the things that I do need to pay attention to, like if something happens to my daughter or her friend, or if they need me, or if they call, that I would get enough of a sensation, so there would be enough action potentials produced, that they would get sent to my brain then would be able to interpret them as not only a sound, but as a specific sound coming from either my daughter or her friend that they needed something. And then I would be able to do something about it. So hopefully the difference between sensation and perception makes sense, but also hopefully what makes sense is that we don't experience everything that's happening in the world. There's no possible way we can. There is too much happening, too many sights, too many sounds, too many smells, too many touches. I mean, if you think about it right now, if you're wearing clothes, and if you're not, please don't tell me, right? But if you're wearing clothes, right, every single moment that your clothes are touching your skin, technically, right, your body is probably sending a message to your brain saying, I got clothes on, I got clothes on, I got clothes on, I got clothes on. But eventually, one or two things, or maybe even both, are going to happen. Right? Either the sensory organs are going to stop sending that information, or your brain is going to start to ignore that incoming information because it hasn't changed in a while. But we'll talk about that later. Why? Because that information is no longer important. I don't need my brain to keep registering that I have clothes on. Unless something weird changes, like, wait a minute, why am I wet? Or why did I get cold all of a sudden? Or it feels like I have a hole in my shirt. Right? In that case, I've gotten a new sensation that I have then interpreted. Right? The new sensation got sent to the brain that I then interpreted as, wait a minute, that feels like a hole. That's not good. That isn't what I should normally have. And then I'm probably going to pay attention to it. But if nothing has changed, there's no incoming new information. If I haven't perceived any new incoming information, I'm probably going to ignore it. All right, I know I went on for that for a little bit long, but I want to make sure you get that idea. You don't usually have a conscious acknowledgement of sensation. Sensation is just your eyes, your ears, your nose, your tongue, your skin, converting some kind of energy into an action potential. The only part you actually consciously experience is the perception part. And again, there's multiple layers to that perception, to that interpretation part. All right? For vision, this is a picture of my daughter quite a number of years ago. Here's sort of what's happening. When you're looking at something, here's kind of what's going on. Right? Your eyeballs, you have a bunch of different cells in your eyeballs. Each one of them, right, will be sending an individualized message to the brain. Right? Because it will have been struck by a different amount of light, a different combination of incoming light energy. Right? And it will send a message to the brain because it was able to produce an action potential because it was struck by a different combination of light. Right? And then what your brain does is take all of those little bits and pieces of information, those incoming pieces, those incoming action potentials, and put it all together and have it make sense. So your brain is what's responsible for integrating all of the information and making it make sense. So that's sort of what's going on in our nervous system. A little bit more about sensation. Sensation is just when some sort of physical stimuli from the environment interacts with a sensory organ. And again, that can be in your eye, that can be in your ear, it can be in your nose. Right? Again, this is accomplished by the sensory receptors. They are able to detect and then convert that energy, from the environment into a message that's sent to your brain. So you've got a sensory receptor that detects the energy. Each sensory receptor has some kind of specific receptive field. So the cells in your eye have a receptive field in which if there's light within that field, it's going to hit that cell in your eye. You've got a receptive field where if there's sound waves in that field, it's going to go into your ear. You have on your skin a particular receptive field where if something touches your skin in that area, it will set off an action potential for that cell. All right? So that sensory information will then be encoded. In other words, it will produce an action potential. Right? And that action potential will then get to the brain via neural relay, which means it will get sent from neuron to neuron to neuron, just as we talked about before. So you have action potential, gets to the end, releases neurotransmitter, which stimulates the next neuron, produces an action potential, gets to the end, releases neurotransmitter until it gets to the brain. And then once it's in the brain, That incoming sensory information is then translated into something meaningful, and that's the part that we call perception. Again, I'm saying it over and over again because I want to make sure you have it make sense. So again, because I've said it a bunch of times, you can guarantee there's going to be some questions about sensation versus perception on the test and the quiz. Okay. I want you to learn this pattern too, because you're going to see this pattern a lot. We're going to talk about this pattern for every sensory system. All right, and it sort of starts at the bottom and works its way up. All right, so work with me down here. So down here is where the sensation part happens. Literally, it happens with these first two steps. So sensation is down here. There's some kind of sensory information, and that sensory information stimulates a receptor. All right, the first one we're going to do is for light and for vision. So the sensory information is going to be light, and it's going to interact with a receptor in your eyeball. And then again, for every kind of sensory system, right, that information is going to be sent from those receptors. Ultimately, it's going to go through the thalamus, right, to a primary sensory cortex. And when it hits this primary sensory cortex, this is when perception starts to happen. This is when your brain starts to go, oh, this is what I'm, in the case of vision, seeing, or in the case of auditory, this is what I'm hearing. Right? And it processes it in a lot of different ways. It puts it together with other sensory information. So for example, you might put together something that you're seeing and hearing as one thing. Oh, so I see that dog over there and I hear it barking. So that the dog over there that I'm seeing is the dog that's barking. And you put it together as one thing. So you're going to see this pattern over and over again. And this is basically going to be the outline of the lectures that we're going to do for the next couple of lectures. Is you're going to hear me talk about what kind of sensory information we're going to be talking about for which sensation, for which sensory system. You're going to hear me tell you about the receptors. You're going to hear me tell you about which part of the thalamus it goes through, and then which part of the brain you find the primary sensory cortex, and then how it's processed by other secondary or association cortex. So we're going to do this pattern a lot. So sensory receptors are specialized cells that transduce. That's a fancy word. That's a cool word. Transduce just means to convert. Right? So it's going to transduce the sensory energy into neural activity. So in your eye, you have specialized cells that convert or transduce light energy into action potential. In your ear, you have specialized cells that transduce. By the time it's gotten there, it's actually moving water or moving liquid into action potential. Again, each sensory system's receptors are designed to respond only to a certain kind of energy. So vision, it's light energy. Auditory information, it actually starts as air pressure, but we call it mechanical energy because, again, it starts as air pressure. That air pressure ultimately moves a membrane called the tympanic membrane. You might call it your eardrum. That then moves a couple little bit of bones, which is why we call it mechanical. It's moving bones in your inner ear, in your middle ear. And then those bones tap on this thing that's filled with fluid in your inner ear, and it causes kind of like ripples, which is still moving water, right, or moving liquid. So it's mechanical energy is what we call it for your ear. Your somatosensory system, your sense of touch, same idea. It's when things touch or move your skin. So we call it mechanical energy. And then for taste and smell, and the fancy word for smell is olfaction, it's also chemical energy. So it's literally chemicals that go into your nose or chemicals that go into your mouth. So what you're tasting when you're eating food or when you're enjoying some candy or something like that is the chemicals that's in the food, the chemicals that's in the candy. And then again, your sensory receptors have a receptive field. They have a specific part of the world to which they can respond to the energy in that part of the world. So the light that's in that part of the world, the moving air pressure, the chemicals, that sort of thing. Right? The more receptors you have, the more sensitive you're going to be. So if you have more receptors in your eye, you're going to be able to see more clearly in that particular part of the world. If you have more receptors on your skin is going to be more sensitive in that area. So if you think about the sensitive parts on your skin, Right? Think about the parts of your skin where you touch stuff and you can tell what stuff is the most. Right? It's probably going to be things like your hands, your face. It's probably not going to be the middle of your back. Now, if you're like me, the middle of your back might be really ticklish, right? But I don't usually use the middle of my back to feel what something is. I don't usually use the middle of my back, right, to figure out what something is. I usually use my hands because my hands have a ton of sensory receptors in them. So they're particularly sensitive, right, and that gives me a lot of information. When it ultimately reaches the brain is able to perceive it. So I can use my hands to figure out a lot of information. Because of that, there's a lot more receptors in my hands and in my face than there are, say, in the middle of my back. Also, different organisms have different sensory abilities. For example, your olfactory system or your ability to smell is a lot less acute than that of some other kinds of animals like dogs or rats, for example. However, your sense of vision is a lot better than that of some creatures, for example, moles or even the rat. So again, we tend to have better sensory systems, or our sensory systems, actually, this is a better way of wording it, are more developed for those that we use and we rely on more. So a dog and a rat rely a lot more heavily on their sense of smell. Humans rely a lot more heavily on our sense of vision than we do a lot of our other sensory systems, or at least we have evolved that way over time to rely more and more and more on our sense of vision. One of my cool random fun facts about dogs and rats, actually both of them can be used to detect bombs. You've probably heard of a bomb sniffing dog or even drug sniffing dogs. Yes, they can be taught, they can be trained, they work with their trainer and they're taught to give a certain signal when they detect explosives and when they detect drugs or whatever it is they've been taught to do. But more recently, they've actually been training rats to do the same thing, because not only do rats have an amazing sense of smell, but they're also super intelligent as well. And they weigh a lot less than a dog does, and they can get into places that are a lot smaller than a dog is. So they can get into places that a dog can't, and they don't usually set off explosives that are weight dependent that a dog probably would, so they don't get injured as readily as a dog would. So they've actually been, the military especially has been training rats to sniff out explosives and give us particular signal when the rat smells the explosive. Again, that's how sensitive their olfactory system is. Well, so again, it just depends on what your body needs and what your body uses. We also see in individuals, and this is within a species now, so within the human species, for example, if somebody is either born without a particular sensory ability or loses a particular sensory ability, they will experience sometimes a heightening of a different sensory ability, or sometimes, for example, if you lose a limb, you have kind of a heightening of sensory ability around where that limb was, or a heightening of a sensory ability in a different location. So again, we have this, our brain is really cool, and I know Dr. Bennett has mentioned this a bunch in some of the other videos. Your brain is what we call plastic. It can change and adapt, and one of the things that it can do is also do that same thing, but with your sensory systems. And so your brain can change and adapt, right, to be able to take in new and different cool kinds of information and adapt to adjusting information, even if your body structure has actually changed as well. Again, we also know, following this sort of pattern that we have talked about, is that all receptors are going to ultimately get to the cortex, right? But there isn't this like straight thing that I talked about. So I showed it to you in this pattern to sort of talk to you about the way that I'm going to be presenting the information. So when I first talk to you about a sensory system, I'm going to show you the receptors, or we're going to talk first about the receptors. Then we'll talk about the pathway to the thalamus. Then we'll talk about how it gets to the primary sensory cortex, and then how it is further processed in the secondary sensory cortex, as well as any association cortices. But what we know is that it's really not that simple. It's not like a 1 to 1 to 1 to 1. It really looks more like this. Let's see if I can't move the picture. All right, so what should be there is a whole jumbled up, so it should look like basically a big plate of spaghetti with a whole bunch of noodles, like all jumbled up, right? Is that there really is no straight through point-to-point correspondence. Let me go see if I can find a picture and show you exactly what I mean by this jumbled picture. The problem is that I'm working with a PowerPoint. I'm not working with a PowerPoint. I'm working with an old set of PDFs, which is what you guys have, to recreate some of these lectures to make them more of how I want them to be. So let me go see if I can find an image of this, and I'll come right back. Okay, so I'm not going to stay on here for long, but hopefully you can sort of get a visual of what I was talking about. This was the old video that Dr. Bennett had put together, but again, you can see there's a lot of crosstalk even within the thalamus. There's crosstalk within the primary sensory cortex. So again, and there's feedback, all kinds of different directions. So again, it's not this pretty picture that I showed you. It is, but it isn't. So don't let this confusion throw you off. What I really want you to focus on for this class is the more 1 to 1 to 1 to 1 picture, and then we will come back later and we will go back and add the other context to it, again, especially if you go on to take the next class, which was 3113 physiological psych. Okay, so hopefully that made sense. Again, don't let yourself get tripped up by the reality of the situation, which is that it really is a lot more complex than just this 1 to 1 to 1 pattern. Okay, because in the very next lecture, so again, this is basically the end of this first lecture here for Unit 2, but where we are headed from here, right, is we are going to start the visual system. So I am going to end this lecture. I'm going to show you one final thing in this lecture, which is a crash course review video about sensation and perception. Again, one more time to drive home that concept. But then where we will pick up in the next video is actually right here on this slide. So we're going to start with the visual system, and we're going to talk through, again, this idea of what are the receptors? Where are they located? Well, hopefully it's very obvious for you that in the visual system, the receptors will be located in the eye. We'll talk about how the eyeball works, all the cool parts of the eyeball, and then how that information gets sent to the thalamus, which specific part of the thalamus, how that information gets sent to the primary sensory cortex. And if you were at all paying attention in Unit 1, you will know that the primary sensory cortex is the occipital lobe. We'll talk a little bit more about the occipital lobe and then how it sends that information to some other parts of the brain for some very cool processing that allows you to see all the cool stuff that you can see. So that's where we're going to be headed in the very next lecture. We'll talk about how you can see things in color and all kinds of cool stuff like that. But again, I'm going to end here as far as the slides go for this particular lecture, and I will end with a crash course video about sensation and perception. So I will see you guys soon.
Let me tell you about Oliver Sacks, a famous physician, professor, and author of unusual neurological case studies. We'll be looking at some of his fascinating research in future lessons, but for now, I just want to talk about Sacks himself. Although he possesses a brilliant and inquisitive mind, Dr. Sacks cannot do a simple thing that your average toddler can. He can't recognize his own face in the mirror. Sacks has a form of prosopagnosia, a neurological disorder that impairs a person's ability to perceive or recognize faces, also known as face blindness. Last week we talked about how brain function is localized, and this is another peculiarly excellent example of that. Sacks can recognize his coffee cup on the shelf, but he can't pick out his oldest friend from a crowd because the specific sliver of his brain responsible for facial recognition is malfunctioning. There's nothing wrong with his vision. The sense is intact. The problem is with his perception, at least when it comes to recognizing faces. Prosopagnosia is a good example of how sensing and perceiving are connected, but different. Sensation is the bottom-up process by which our senses like vision, hearing, and smell receive and relay outside stimuli. Perception, on the other hand, is the top-down way that our brains organize and interpret that information and put it into context. So right now, at this very moment, you're probably receiving light from your screen through your eyes, which will send the data of that sensation to your brain. Perception, meanwhile, is your brain telling you that what you're seeing is a diagram explaining the difference between sensation and perception, which is pretty meta. Now, your brain is interpreting that that light as a talking person, whom your brain might traditionally recognize as Hank. We are constantly bombarded by stimuli, even though we're only aware of what our own senses can pick up. Like, I can see and hear and feel and even smell this corgi, but I can't hunt using sonar like a bat or hear a mole tunneling underground like an owl or see ultraviolet and infrared light like a mantis shrimp. I probably can't even smell half of what you can smell. No. We have different senses. Yeah. There's a lot to sense in the world, and not everybody needs to sense all the same stuff. So every animal has its limitations, which we can talk about more precisely if we define the absolute threshold of sensation, the minimum stimulation needed to register a particular stimulus 50% of the time. So if I play a tiny little beep in your ear, and you tell me that you hear it 50% of the times that I play it, that's your absolute threshold of sensation. We have to use a percentage because sometimes I'll play the beep and you'll hear it, and sometimes you won't, even though it's the exact same volume. Why? Because brains are complicated. Detecting a weak sensory signal like that beep in daily life isn't only about the strength of the stimulus. It's also about your psychological state, your alertness and expectations in the moment. This has to do with signal detection theory, a model for predicting how and when a person will detect a weak stimuli, partly based on context. Exhausted new parents might hear their baby's tiniest whimper, but not even register the bellow of a passing train. Their paranoid parent brains are so trained on their baby, it gives their senses a sort of boosted ability, but only in relation to the subject of their attention. Conversely, if you're experiencing constant stimulation, your senses will adjust in a process called sensory adaptation. It's the reason that I have to check and see if my wallet is there if it's in my right pocket, but if I move it to my left pocket, it feels like a big uncomfortable lump. It's also useful to be able to talk about our ability to detect the difference between two stimuli. I might go out at night and look up at the sky, and well, I know with my objective science brain that no two stars have the exact same brightness. And yeah, I can tell with my eyeballs that some stars are brighter than others. But other stars, they just look exactly the same to me. I can't tell the difference in their brightness. Are you done? Is it time for you to go? Gimme a yes. Okay. Good girl. The point at which one can tell the difference is the different threshold. But it's not linear. Like, if a tiny star is just a tiny bit brighter than another tiny star, I can tell. But if a big star is that same tiny amount brighter than another big star, I won't be able to tell the difference. This is important enough that we gave the guy who discovered it a law. Weber's law says that we perceive differences on a logarithmic, not linear scale. It's not the amount of change, it's the percentage change that matters. All right, how about now we take a more in-depth look at how one of our most powerful senses works. Vision. Your ability to see your face in the mirror is the result of a long but lightning-quick sequence of events. Light bounces off your face and then off the mirror and then into your eyes, which take in all that varied energy and transforms it into neural messages that your brain processes and organizes into what you actually see, which is your face. Or if you're looking elsewhere, maybe you see a coffee cup or a corgi or a scary clown holding a tiny green pie. So how do we transform light waves into meaningful information? Well, let's start with the light itself. What we humans see as light is only a small fraction of the full spectrum of electromagnetic radiation that ranges from gamma to radio waves. Now, light has all kinds of fascinating characteristics that determine how we sense it. But for the purposes of this topic, we'll understand light as traveling in waves. The wave's wavelength and frequency determines their hue, and their amplitude determines their intensity or brightness. For instance, a short wave has a high frequency. Our eyes register short wavelengths with high frequencies as bluish colors, while we see long, low-frequency wavelengths as reddish hues. The way we register the brightness of a color, the contrast between the orange of a sherbet and the orange of a construction cone, has to do with the intensity or amount of energy in a given light wave, which, as we just said, is determined by its amplitude. Greater amplitude means higher intensity, means brighter color. Someone just told me that sherbet isn't a word that exists. His name is Michael Aranda, and he's a dumbhead. Did you type it into the dictionary? Type it into Google. Ask Google about sherbet. So sherbet is a thing. So after taking in this light through the cornea and the pupil, it hits the transparent disc behind the pupil, the lens, which focuses the light rays into specific images. And just as you'd expect the lens to do, it projects these images onto the retina, the inner surface of the eyeball that contains all the receptor cells that begin sensing that visual information. Now, your retinas don't receive a full image like a movie being projected onto a screen. It's more like a bunch of pixel points of light energy that millions of receptors translate translate into neural impulses and zip back into the brain. These retinal receptors are called rods and cones. Our rods detect grayscale and are used in our peripheral vision, as well as to avoid stubbing our toes in twilight conditions when we can't really see in color. Our cones detect fine detail and color. Concentrated near the retina's central focal point called the fovea, cones function only in well-lit conditions, allowing you to appreciate the intricacies of your grandma's china pattern or your uncle's sleeve tattoo. And the human eye is terrific at seeing color. Our difference threshold for colors is so exceptional that the average person can distinguish a million different hues. There's a good deal of ongoing research around exactly how our color vision works, but two theories help explain some of what we know. One model, called the Young-Helmholtz trichromatic theory, suggests that the retina houses three specific color receptor cones that register red, green, and blue, and when stimulated together, their combined power allows the eye to register any color. Unless, of course, you're colorblind. About 1 in 50 people have some level of color vision deficiency. They're mostly dudes because the genetic defect is sex-linked. If you can't see the Crash Course logo pop out at you in this figure, it's likely that your red or green cones are missing or malfunctioning, which means that you have dichromatic instead of trichromatic vision and can't distinguish between shades of red and green. The other model for color vision, known as the opponent process theory, suggests that we see color through processes that actually work against each other. So some receptor cells might be stimulated by red but inhibited by green, while others do the opposite, and those combinations allow us to register colors. But back to your eyeballs. When stimulated, the rods and cones trigger chemical changes that spark neural signals, which in turn activate the cells behind them, called bipolar cells, whose job it is to turn on the neighboring ganglion cells. The long axon tails of these ganglions braid together to form the ropey optic nerve, which is what carries the neural impulses from the eyeball to the brain. That visual information then slips through a chain of progressively complex levels as it travels from optic nerve to the thalamus and onto the brain's visual cortex. The visual cortex sits at the back of the brain in the occipital lobe, where the right cortex processes input from the left eye and vice versa. This cortex has specialized nerve cells called feature detectors that respond to specific features like shapes, angles, and movements. In other words, different parts of your visual cortex are responsible for identifying different aspects of things. A person who can't recognize human faces may have no trouble picking out their set of keys from a pile on the counter. That's because the brain's object perception occurs in a different place from its face perception. In the case of Dr. Sachs, his condition affects the region of the brain called the fusiform gyrus, which activates in response to seeing faces. Sachs's face blindness is congenital, but it may also be acquired through disease or injury to that same region of the brain. And some cells in a region may respond to just one type of stimulus, like posture or movement or facial expression, while other clusters of cells weave all that separate information together in an instant analysis of a situation. That clown is frowning and running at me with a tiny cream pie. I'm putting these factors together. Maybe I should get out of here. This ability to process and analyze many separate aspects of the situation at once is called parallel processing. In the case of visual processing, this means that the brain simultaneously works on making sense of form, depth, motion, and color. And this is where we enter the whole world of perception, which gets complicated quickly and can even get downright philosophical. So we'll be exploring that in depth next time, but for now, if you were paying attention, you learn the difference between sensation and perception, the different thresholds that limit our senses, and some of the neurology and biology and psychology of human vision. Thanks for watching this lesson with your eyeballs, and thanks to our generous co-sponsors who made this episode possible: Alberto Costa, Alpna Agrawal, PhD, Frank Zegler, Philip Detmer, and Kurzgesagt. And if you'd like to sponsor an episode and get your own shout-out, you can learn about that and other perks available to our Subbable subscribers. Just go to subbable.com/crashcourse. This episode was written by Kathleen Yale, edited by Blake DiPastino, and our consultant is Dr. Ranjit Bhagwat. Our director and editor is Nicholas Jenkins. The script supervisor is Michael Aranda, who is also our sound designer, and our graphics team is Thought Cafe.